I'm sorry, I'm a little bit sick, so I will cough every word probably. Well, anyway, we'll have today webinar by Mattia Pelizzola from Italy, Institute of Italiano and the Tecnologia, who is going to talk about specific technology, Nanodynamo. Uh, uh, he will explain how this technology works for studying RNA metabolism. And then we'll have his postdoc, another Mattia Furlan, who's going to talk about this technology, but applied to breast cancer and drug treatment to this pathology. So I guess this is the only thing I'm going to say. And so Mattia, if you wanna, well, both of you call Mattia, but Mattia Pelizzola, if you wanna start the introduction, go ahead.
Okay, thanks a lot for the opportunity, for the invitation. So I will go ahead and share mine. Okay. So yes, today we will talk about dynamics of RNA metabolism. In my introductory shorter presentation, I will try to describe what we mean by this term, and then I leave the stage to Mattia for the actual real scientific presentation. So in general, in our group, we apply computational and experimental approaches to understand how common post-transcriptional events shape gene expression programs. Okay, so in this outline presentation, this introductory presentation, we'll cover the concept of dynamics of RNA metabolism. I will outline, illustrate very briefly an application of it, and then I'll leave the stage for going into the details of Nanodynamo by Mattia Furlan. So RNA dynamics, what do we mean by that? The general concept is that even though you can find RNA molecules from cells, from a population of cells, this does not automatically imply that the corresponding gene is actually active, as maybe is typically considered. Maybe the gene in the meanwhile has been silenced, maybe epigenetically, and the RNAs are still floating in the cells. As another example, I'll try to activate the pointer. Okay. As another example, if you find that there is an increased number of RNA molecules over time or across conditions, this does not automatically imply that the corresponding regulation is transcriptional in nature, meaning an increase in the rate of synthesis of novel RNA. Maybe it's just because there is a stabilization and a reduced rate of degradation of those RNAs, and these RNAs are just accumulating there because they are degraded less efficiently. So it's very contrasting and different molecular mechanisms that can generate very similar outcomes in terms of transcription. So how to disentangle? Of course, it's quite important to try as much as possible to decipher what's going on, because this also offers specific possibilities for then intervening and potentially changing that, especially in case of diseases. So the way that we are doing it is to apply computational modeling approaches based on differential equations and mathematical modeling. Initially, we used this quite simple framework to describe our transcriptional system. We focused on the key steps of RNA synthesis, so production of new immature molecules of RNA that eventually is processed and spliced into the mature form that eventually is degraded. So we start for a few years to playing with these individual steps: synthesis, processing and degradation. And we describe them using mathematical modeling, as I said, and basically is about writing equations that describe how the amount of either premature or mature forms are built up and maintained into the cellular system by the contrasting action of different forces. So premature RNA is produced by the synthesis of another RNA and is subtracted from the system by its conversion into another form, the mature form, through the step of RNA processing, while the mature form is the balancing between its creation through the processing of the immature one minus its degradation through the force of degradation. At steady state, basically, the change of this of the abundance of either premature or mature RNA over time or across conditions is basically zero. Everything is flat over time. So basically, this implies that premature RNA is determined as the balance between synthesis and processing, and mature RNA abundance is determined by the balance between synthesis and degradation. Over time, when things are changing across time or across conditions, things start to be more, let's say, interesting. For example, very naively, this is based on simulated data for the moment. When there is an increase in the rate of synthesis, what's happening is obviously that there is an amount of premature RNA that is increasing, closely followed by an increase in the mature form over time. If you double the amount of the rate of synthesis, you will get twice the amount of the abundance of both species. If there is an increase in the rate of processing, then what's going to happen is that there is a decrease in the amount of premature RNA because this is converted more efficiently, while there is no change at the level of the mature. Conversely, if there is a decrease in the rate of the degradation, there is an accumulation of the mature RNA, while the premature RNA is not changed. If this decrease in degradation, for example, as you can see here again, is coupled together with an increase in synthesis, then these two parallel modulations will strengthen each other, and you will have even further accumulation of the mature RNA. While if they are modulated in the same direction, basically they contrast to each other, and the final result is basically that the mature RNA is not modulated anymore because it's created more, but is also degraded more. So these are just examples to see how different mechanisms can generate quite interesting and complicated patterns in terms of premature and mature RNA. Now, I introduced a mathematical model system that can be used to model, to reason, to quantify all these things. But in practice, these systems cannot be solved with typical data because it is undetermined, meaning that you have three unknowns, the three kinetic rates of synthesis, processing, and degradation, and only two pieces of data, the abundance of premature and mature RNA that you can actually sequence. So what you miss is an extra ingredient, and there are different options here. We might have a proxy for the rate in which you provide drugs that block transcription, and then you follow the exponential decay of RNA species over time, getting quantitative information about the half-life of those transcripts. But this is a super strong perturbation on the cellular system, so it's very invasive and it's time-consuming. As an alternative, in the last years, RNA metabolic labeling has established as a very good idea. It provides a very good proxy of RNA synthesis. It works so that you can provide to the system modified nucleotides. It can be 4SU or 5U that get incorporated from the medium into the RNAs that are transcribed, typically during a certain amount of time, let's say half an hour or 1 hour or 2 hours. And the labeled RNAs are those that are nascent, that have been transcribed during this specific amount of time. The impact on cell metabolism is minimal or none. Still, it requires twice the sequencing because typically you need to then purify the nascent population and sequence it apart from the pre-existing one. So there has also been further evolution in these technical directions through chemical derivatization approaches, in which the labeled RNAs, the modified nucleotides, is actually converted into different bases that can be identified through the sequencing. Yet this is typically affected in terms of sensitivity and typically requires a longer pulse. It cannot be adapted very easily for like a few minutes pulses. But in general, metabolic labeling prevailed in terms of offering a very important piece of data for this kind of analysis and providing, very importantly, a proxy for the rate of RNA synthesis. So eventually, we developed a number of tools which fall out and inspect umbrella of tools is an bioconductor package that offer the possibility to explore how synthesis, processing, and degradation are quantified and behave at steady state, or also how they vary across conditions or over time. And we even developed a solution in which actually metabolic labeling is not even required, but you do need to profile a time course experiment with several data points, at least 2 different time points. And we also developed a graphical interface in which you can play and try to understand how the combination of different kind of regulations, transcriptional and post-transcriptional, eventually impact on the temporal shapes of the transcriptional evolution of both premature and mature RNA forms. On the other hand, I also want to give in just 2 slides an idea that a similar kind of problems, we also we have exactly the same issue in terms of the polymerase itself. So RNA polymerase 2, that we are all familiar with, the interpretation of the data about the abundance and the binding of this complex is also affected by similar issues. Is also undergoing a life cycle where the polymerase is recruited. Then there is the step of pores release, elongation, and detachment, right? And also for polymerase, there is a similar problem. So when there is an increase in polymerase at the promoters, is this due to faster recruitment? Or is this due to slower post-release, for example? Different mechanisms that can result in the same identical outcome. So how it is out this, that of course is very much intertwined and linked with the transcriptional program. We developed a very similar solution based on the same concept, where the abundance of promoter at each step, either promoter or gene body or triple strands, is regulated by the balancing of contrasting forces, the recruitment and the post-release, and then the movement of pull-2 during the elongation in the gene body. And eventually we have been able to measure quantify the specific rates of post-release, elongation, detachment, and recruitment. Again, genome. Okay, I keep going. I hope you can still hear me well.
I think the problem was me, so sorry for that.
Okay, no problem. So what we've been doing here is that we acutely activated this MYC transcription factor, and we follow the transcriptional response over time for several time points. We profiled mature and RNA and premature RNA over time, so genes that are induced or repressed following the activation of MYC. And through the metabolic labeling, as I told you before, we also profiled nascent RNA specifically with the labeling of 10 minutes, I think, in this case. And then through our mathematical modeling, we basically converted this data into a more complex information, more comprehensive information, where premature RNA were associated with the modulation of the rate of synthesis, processing, and degradation, showing that, as expected, MYC modulation has a major impact in terms of transcriptional regulation synthesis of the target genes, but also 1/4 and 1/3 of the target genes were modulated in terms of the efficiency of processing and degradation. In parallel, we have been also profiling the polymerase by ChIP-seq at the promoters, gene body, and transcription and site, genes on the rows and cluster of genes specifically. And through the modeling that I shown to you before, we converted this data into more informative quantification of the variation of the rate of recruitment, post-release, elongation, and detachment of the polymerase complex. For example, these are genes that are increased in recruitment, increased in post-release, and decreased in elongation over time following MYC activation. And just to give a final example here, these genes, for example, those on this red box, are genes where polymerase is increasing a lot in the abundance on the gene body. So I think most of us will think that, okay, there is an increased POL2 in the gene body, then is a transcriptional activation, right? It's not really the case, because indeed the synthesis rate hasn't increased yet, it's still quite flat. So this is not productive POL2 that is binding there, but is POL2 that is accumulating there, is in a non-further productive state. Indeed, our mathematical model interpreted this as a reduced rate of elongation of the complex that created this as if it is a traffic jam of unproductive polymerase. And eventually, we found out that this is a transient dip that occurs during the 3-4 hours, the first 3-4 hours of the MYC acute activation, in which we have a strong reduction in terms of elongation of the polymerase, and concomitant to a strong reduction in the rate of processing. Altogether, this is causing an accumulation of pre-RNA, premature RNA, and only after like 4 or 5 hours, the proper transcriptional response starts, and you see a quite diffuse and strong increase in the rate of RNA synthesis. And at this point, this transcriptional burden block, let's say, is resolved, and the proper result of and expected result of the MYC activation starts. Just to give an example of the things that you can learn through this kind of data acquisition and analysis. So with this part, we have shown that the acute activation of MYC markedly impacts RNA dynamics, not just in terms of synthesis of the target genes, and that how it is important to study RNA dynamics for understanding transcription factor-driven gene expression programs. And I will now leave the stage to Mattia, because, of course, reasoning in terms of synthesis, processing, and degradation can be fun, can be interesting, but of course, just the tip of the iceberg of the RNA life cycle, right? Plus, we are in Translacore, so we need to speak about translation somehow. And we will come to that in the next part. I will now switch to Mattia's presentation. Here we go. Thank you, Mattia. And thank you everyone for being here, for the opportunity to present this work, which is about Nanodynamo, our latest research project in the field of RNA dynamics. So I will open my presentation with just one slide to explain you the reason why we decided to start the Nanodynamo project. Then I will present you the experimental and computational framework. I will tell you something about its validation and its application to investigate triple negative breast cancer.
And finally, I will spend a few slides on ongoing projects, very preliminary results from ongoing projects, which are somehow the follow-up of Nanodynamo. Okay, so as Mattia said, RNA dynamics are a very powerful perspective to investigate transcriptional programs, but these methodologies are affected by a severe limitation, which is the poor resolution. Indeed, if you think about INSPECT, as Mattia said, in this framework, the RNA life cycle is a very simple 3-steps process, which accounts for RNA synthesis, RNA processing, and RNA degradation. But we know that much more is going on. For instance, we know that in eukaryotic cells, transcripts are synthesized into the nucleus and are usually exported into the cytoplasm, where they can engage with the translation machinery. So 2 very important steps that were missing from our previous models. And of course, one of the main ideas behind Nanodynamo was indeed to extend the resolution to investigate the novel processes included into our model of gene expression. But not just as standalone processes. Also, Nanodynamo was meant also to get a full picture in a given biological system of gene expression at this resolution in order to investigate the interplay between different steps of gene expression. And this is very relevant. For instance, in this cartoon, you see effectors that are already known to be involved in the coupling between translation and many other steps of gene expression, like transcription, processing, export, and degradation. So the idea with Nanodynamo was to implement this novel framework in order to investigate gene expression at higher resolution and in order to study simultaneously the regulation of different steps of gene expression in order to reveal couplings. Okay, so now that the aim is clear, let's move on with a description of the framework. Okay, here you see the novel model of gene expression that we implemented into Nanodynamo. So we start with the synthesis of chromatin premature RNA, which can be immediately spliced to become chromatin mature RNA. And later, chromatin mature RNA can be detached to enter the nucleoplasm as nucleoplasmic mature RNA. On the other hand, premature molecules can be detached in an unspliced form to become nucleoplasmic premature transcripts, which can later be post-transcriptionally processed. Nucleoplasmic mature RNA can be exported in the cytoplasm, where it can be immediately degraded, where it can be associated with polysomers and later degraded. So you can appreciate from this cartoon the higher resolution that Nanodynamo provides in terms of description of gene expression. Okay, here you see the equations, which are the mathematical formulation of this model. But more important than equations here are the RNA species involved in the model and therefore needed to fit the model. So we have chromatin and nucleoplasmic premature and mature RNA, cytoplasmic RNA, and polysomal RNA. And we need both pre-existing and nascent RNA for all these RNA species. So how can we collect this amount of information? In this way, we start with metabolic labeling, and then we isolate chromatin, nucleoplasmic, cytoplasmic, and polysomal RNA. We prepare libraries for direct RNA sequencing, and we sequence. And after sequencing in silico, we can detect premature reads according to the intronic signal, and nascent reads according to the presence of FORCE. And I will spend a few words on this step later in the second half of the presentation. So with this experimental framework, we can collect all the information needed to fit our model. But is it enough? So given this information, are we able to really perform our inference task? To answer this question, we created simulated datasets where we know the real kinetic rates, of course, and we can try to recapitulate them. So here you see all the kinetic rates from synthesis down to polysomal degradation. And in each box, you have the expected kinetic rates here and the inferred ones on the other axis. And within each box, we also reported the Spearman correlation coefficient. So looking at these clouds and at the numerical values, we can really conclude that indeed we are able to perform our inference task. Then we used simulated data also to define the optimal experimental design to apply Nanodynamo in order to investigate our systems of interest. Specifically, we created simulated datasets with a variable number of labeling pulses, because of course one is needed, but you can decide also to have more than one. So here you see 20 minutes plus 1 hour, 20 minutes plus 1 hour and 2 hours of labeling. And we investigated how this increased information provided by the additional labeling pulses improves the inference performance. Here again, you see the Spearman correlation coefficient for all the kinetic rates. And you see that somewhere you have the expected increase in performance given the additional labeling pulses, but this is not always the case. Okay. We follow the very same approach also to investigate the optimal number, to identify the optimal number of replicates. So creating simulated datasets from 1 up to 5 replicates. And again, here you see more systematic increase in inference performance given additional replicates. So at the end of the day, looking at these bar plots, we decided that 1 labeling time and 2 replicates could be a good trade-off between experimental load, cost, and quality of the inference. And we decided to apply this experimental design, to provide RNA dynamics with Nanodynamo in triple-negative breast cancer cells, some 159 cells. So from this slide on, you will see all the results that you will see are based on real experimental data. Okay. So in this slide, I decided to recapitulate for one gene, quite relevant in the context of triple-negative breast cancer, which is GSTP1. So I decided to recapitulate what you get from the experimental framework of Nanodynamo and from the inference one, the computational one. So on the left here, you see all the RNA species that we collect with our experimental framework. And here you see the full set of kinetic rates that we infer. The synthesis rate K1 has been depicted in gray because it cannot be directly compared with the others. But of course, you can compare synthesis across genes. So this is still relevant. Okay, so here you see the result of Nanodynamo in terms of kinetic rates for this gene. But how can we interpret these numbers? In the following way. So Nanodynamo is telling us that GSTP1 is efficiently co-transcriptionally processed, while the post-transcriptional processing branch is less efficient, specifically due to a low rate of detachment of premature molecules. Then mature nucleoplasmic transcripts are efficiently exported in the cytoplasm, where they tend to be associated with poisons and later degraded. So this is the description of GSTP1 transcription in some 159 cells. Of course, you can do the very same analysis for thousands of genes, and here you see the distributions of the kinetic rates, again, from synthesis down to polysomal degradation, for all these genes that we profiled with our genome-wide study. So looking at these distributions, we can immediately see that specific rates have specific order of magnitudes. I mean, there are faster and slower rates. Moreover, we can see that there are distributions which are narrow and others that span several orders of magnitude. So the first are probably more tightly regulated by the cell, while the others are more involved in the definition of heterogeneous transcriptional programs for different genes. And we can also appreciate that there are several bimodal distributions, which of course means that for a given rate, there is a set of genes which are more efficiently, in this case, detached, while others are less efficiently detached. This can be appreciated also from this heat map. Here I reported the genes on the rows, while on the columns you have expression levels and kinetic rates. And each quantity is normalized between 0 and 100. So GSTP1 is somewhere here. It is efficiently transcribed and highly expressed in this line. We can see that highly expressed genes require also an efficient synthesis rate and are associated with efficient co-transcriptional processing and inefficient post-transcriptional processing. But this is not always the case. Indeed, here you see a set of genes where the co-transcriptional processing rates are very low, while the post-transcriptional counterparts are very high, very fast. And these two processing branches are mutually exclusive. This is something interesting and unexpected, at least to us, that we further explored. So, Nanodynamo already revealed interesting features of some 159 transcriptional programs. But to go further into our study, we decided to investigate the response of this cell line to a set of perturbations which affect key steps of gene expression. So we started with a drug called Pladinolid B, which is a splicing inhibitor. And as you can see from these genome browser tracks, compared to the untreated cells, after the Pladinolid B treatment, we have a strong increase in intronic signal. And this is true, of course, genome-wide. Here, I reported the proportion of premature reads for untreated and Pladinolid-treated cells. And you can appreciate here the shift of the distribution, which means, of course, that the amount of premature reads is increasing. Okay. The question is, how are the cells responding to this perturbation? So in this heat map, you see, again, genes on the rows, while on the columns you have, again, expression levels and kinetic rates. But this time, you are not looking at absolute values, but you're looking at the modulation of these quantities in response to Pladinolid B compared to the untreated counterpart. Specifically, red means that the quantity is going up, and blue means that it's going down. So, looking at this heat map, we can see immediately that premature species, so chromatin nucleoplasmic premature RNA, is going up, as expected due to the effect of the drug. And the reason is that K2 and K5, which are the splicing rates, are globally going down. However, this is not always the case, because there are sets of genes here and here, which switch from an efficient post-transcriptional processing to an efficient co-transcriptional one, and vice versa. We can also notice a strong downregulation of the synthesis rate, a global downregulation of the synthesis rate, which is the reason why we see all these this downregulation of all the immature RNA species. Moreover, we can appreciate an increase in export, heterogeneous modulations in the cytoplasmic degradation and the association with polysomes, so K7 and K8, and also a global strong increase in polysomal degradation. So here we are looking at a perturbation that affects a specific step of gene expression, splicing, but the result is a very extensive modulation affecting all the steps of gene expression that we have modeled with Nanodynamo. Okay, so let's move to the second treatment, that we apply to our cells, which is the inhibition of proteins export through leptomycin B. Okay, here you see a set of genes that are known to be modulated in terms of export of their transcript in response to the treatment. Specifically, 4 genes whose transcripts are more nuclear retained after leptomycin B treatment, and 2 genes whose transcripts are less nuclear retained. So this is the validation we acquired for our treatment. What about the response of someone under 59 to this perturbation? Okay, here you see the very same heat map I've shown you before, but it is just rotated. So again, you have fold changes, you have expression levels here on the bottom and kinetic rates here on the top. On the left, you have a set of genes which are strongly downregulated in their export, while on the right you have the opposite, genes that are strongly upregulated in their export, in the export of their transcripts. And looking at these heat maps, we can learn, for instance, that genes downregulated in case 6 in export are also downregulated in the rate of synthesis here, and are enriched for genes that are switching from a co-transcriptional processing to a post-transcriptional. Moreover, looking at both these heat maps, you can see that the modulations of K6 are positively correlated with modulations of K7 and K8. So degradations, cytoplasmic degradation, and association with polysomes. Okay, so these are genes that are affected in export, but we can look also at the global modulations for all the genes profiled in this condition. And we can see that there is a strong downregulation of almost any RNA species, and particularly the cytoplasmic ones. And this is associated with a reduction in RNA synthesis. We can again appreciate these processing switches, a global induction of polysomal degradation here, and marked alterations in K7 and K8. So again, perturbation of export and widespread response involving many different steps of gene expression. Finally, we decided to play a little bit also with translation, inhibiting translation with arentonin. Here you see the polysomal files. And here you see a quantification of the RNA hid for polysomal fractions. And you see, of course, the reduction upon treatment with arentonin. This is a problem because, unfortunately, we could not proceed with library preparation and sequencing for arentonin-treated cells. And indeed, we had to implement a simplified model lacking the polysomal RNA quantification, and therefore lacking the association with polysomes and the polysomal degradation. Let me spend just a few words on this concept of simplified models. So we have a full model and a simplified one developed for the arentonin, but we reason that this is not the only case that it's worth it to accommodate, since Nanodynamo requires a lot of information, and this is not always easy to collect. Therefore, at the end of the day, we validated 6 models of different complexity. Here you see the required RNA species, which, of course, provide up to 9 rates with a full model, otherwise provide a subset of the kinetic rates. So if you're interested in this kind of approaches, but you are a little bit afraid of the amount of information needed to fit the full model that we implement in Nanodynamo, keep in mind that we have also these simplified versions. Okay, this is enough for the simplified models. Let's go back to the response to the arentonin treatment in someone under 59 cells. And here we go. So this time, what can we see? Again, we have a reduction in RNA synthesis, and we have switches between co and post-transcriptional processing, so here and here. Indeed, these are matched with an increase in chromatin premature RNA, strong increase in chromatin premature RNA. We have also globally reduced export, and the most affected genes are enriched in those that are switching from co to post-transcriptional processing. And this time, we have a reduced degradation rate. So this is, again, an example of how a perturbation that is affecting translation in very quickly can have important effects also on upstream steps of gene expression. Okay, so I mentioned several times this concept of co-regulations, directly or indirectly, during the initial part of my presentation. Now I will show you what we have done to systematically address this point. So we got interested in this concept of co-regulation of different kinetic rates, close and distal ones. So we decided to start from our log-to-fold change heat maps, or matrices, of course. And we decided to perform a very simple correlative analysis in order to identify a couple of rates which are positively correlated and a couple of rates which are negatively correlated. And here you see the result. This result has been obtained focusing on the response to pladinolide B. Here you see on the nodes of this network, you see the different kinetic rates. And each edge is a significant correlation between the two rates. The color of the edge is indicates the sign of the correlation. So positive correlations in red, negative correlations in blue, while the thickness of the edge is proportional to the strength of the correlation. Again, this is the result for the pladinolide B treatment. We have done the same also for the other two treatments that I discussed before. And here you see the final model. So here you see only the edges that have been confirmed by all our analysis. On the top, you see the nucleoplasmic rates. On the bottom, you see the cytoplasmic ones, and here in the middle, there is the export. What can we see? We can immediately mention this clique here, this positive clique, which involves RNA synthesis and co-transcriptional processing rates. Post-transcriptional processing rates are positively correlated as well, and are strongly and negatively correlated with their co-transcriptional counterparts. And this is expected since we mentioned the mutual exclusivity of these pathways and so on. We can also see this nice clique, which involves synthesis, export, and polysomal degradation. Again, positive correlations between these kinetic rates. And this was expected from the literature, even if we were not sure about the involvement of polysomal, cytoplasmic, or both the degradation rates. Because this is the very first time we have been able to deconvolve the effect of these two degradation steps. Okay, what about the cytoplasmic degradation? You can see this coordination with the co-transcriptional processing, while the polysomal association rate here, K8, is negatively associated with the RNA synthesis and polysomal degradation rate. And again, this edge is was more or less expected. So this is the model, the coupling model that emerged from the application of Nanodynamo to some 159 untreated and treated cells. Then we wondered, but is it true that in order to respond to a stimulus, the cell always exploits all these coupling mechanisms? And to answer this question, we created this heat map. Here you see, again, the genes on the rows. But this time on the columns, you have all the significant edges of the network I've shown you before. So all the significant couplings we detected. And okay, and the entries of the heat map are colored in the following way. So coordinated down regulations, which means that both the rates defining a given coupling are going down, are depicted in green. Coordinated up regulations are depicted in yellow, while opposite regulations, which means that the first rate is going up and the second one down, or vice versa, are depicted in blue. And here, otherwise, you see the absence of coordination, which means that at least one of the rate is not modulated. Okay, so looking at cluster E, and here at the bottom of the heat map, you see that a large fraction of the model genes are responding using almost all the available couplings. And here in the cluster F, you see that many genes that are not regulated in synthesis, because all these couplings here are involving the synthesis rate, are exploiting almost all the post-transcriptional couplings, let's say. While the other half of the map is more sparse. But again, the result is that indeed many couplings are exploited by a significant fraction of the genes responding to the treatment. Okay. Yes, looking at a specific coupling here, like K1 and K2, which is expected to be positive, so populated in green and yellow, we can confirm that most of the genes are regulated in agreement to the expected coupling. However, this is not always the case. For instance, here you see a small set of genes which is not in agreement with our expectation. And this is probably an interesting aspect to further investigate, and that we plan to further investigate. Then we decided to try to identify potential coupling factors, which are responsible for the establishment of these couplings, these co-regulations of rates pairs. And we started looking for RNA binding proteins and transcriptional factors targets among the genes that support a given transcript. And indeed, we found many significant enrichments. Specifically for K1 and K3 and K1 and K4, we found PALBPN1, which is an interesting RNA binding protein involved in the biology of poly-A tails. And we decided to further investigate this potential regulatory layer and potentially coupling layer. And specifically, we profiled poly-A tails length across fractions, several fractions, so chromatin, nucleoplasmic, cytoplasmic, and polysomers. Of course, in untreated, some 159 cells, but also after treatment. And we observed that for the chromatin counterpart, we see a strong reduction in the length of poly-A tails after platinum B treatment, and a significant increase in the length after treatment with leptomycin B and riluzole. So indeed, we are observing a modulation of a global modulation of the length of poly-A tails, which could be interesting to explain some of the results we got, especially the global ones, because these looks like global perturbations. Okay. Of course, all the members of Pelizzola Lab, Mattia, Lucia Valerio, our co-first author of the Nanodynamo paper, current and former members, and also colleagues from BIFO Lab, who help us a lot with polysomal profiling. Also founders, so IARC, the IIT RNA Initiative, and of course Translacore. Here you see an essential bibliography. So this is the Nanodynamo paper. And if you're interested in something about RNA dynamics and RNA modifications, you can take a look at these papers here. And thank you, of course, for the attention.
Okay, thank you, both of you, especially the second Matthias, who took a long time. I don't know if there is some question that the people that are still here would like to ask to Mattia. There is one question in the chat. Okay, and so okay, I cannot open my chat, but maybe you can see it, and maybe you can answer, or
Sure.
you can take time to read it, or
Maybe we can stop sharing and oh no, okay, we can see the chat as yeah. Yes. So let me check the question.
okay.
So I can read it. So
Okay.
how do the
Okay.
6 models of different complexities stack up to one another, and which are the most expendable metrics? And how similar would you expect the models be between cell types? Okay, so regarding the second part of the question, regarding the cell types, I mean, depends on the transcriptional programs. I mean, the point here is to see if different cell types indeed are have constitutive faster, slower rates for specific steps of gene expression. So I think that the answer to this question is indeed the reason why you want to use Nanodynamo in order to get a sense of what are the cell type specific regulations of gene expression. And regarding the 6 model of different complexity, yeah, we compared one again each other for a set of genes that can be modeled with the full one. Then we tried also the simpler one, removing information from for these genes, and I mean they are similar, not identical, of course, for numerical and potentially numerical limitations and so on, but they are similar, they're correlated, of course.
Okay, thank you. I guess, Paula, you have one question because you have your hand left.
Yes. So thank you for these very nice talks from Mattia Pelizzola and Furlan. And I have like 3 questions. The first 2 are about the technique, and then the third one more conceptual. So the first is just because I'm interested. Did you apply this Nanodynamo to yeast cells?
No, we have just applied it to some 159 so far.
Mm-hmm. And about the chromatin RNA purification, how do you fraction this?
Okay, for the technical details about the fractionation protocol, I honestly don't know because it has been done by colleagues in the lab, but the details are in the paper, of course. And if you have any question, please drop us an email. We can share the protocol and so on.
Okay, thanks. And then another question is that if you apply this nanodynamo to a dynamic situation, like for example, during a response to stress in the cells, to see how it changes the regulation to some steps to others?
This is a great question. So this is, of course, a very relevant point, because when you assume that you are observing a modulation, you can investigate it with this kind of approaches that we can call steady state. If your system is sufficiently stable, okay, if you have reached a almost steady state, you can apply Nanodynamo as it is and get a list of kinetic rates. But if you are willing to really follow the modulation in a very detailed way, you have to take into account derivatives, so modulations of the kinetic rates. We have done it with Inspect with a simpler model, so the 3 steps model. So we formulated mathematical functions describing the modulation over time of synthesis, processing, degradation in that case. And in this way, you can get a shape for each rate over time. But right now, this has not been implemented in the Nanodynamo yet.
Mhm.
So inspector, so lower resolution, but already established routine to follow the perturbation over time. Otherwise, you can sample the single point, use the steady state version of Nanodynamo so far, and compare the rates, keeping in mind that there is this limitation.
Okay, thanks. And the third question is, from all the results you have now, you know that it was a theory talking about RNA buffering in the cells in response to different conditions.
Yes.
So do you think that your results support this theory, that if you perturb one step, then there is feedback in others to keep the RNA levels in the cells more or less constant?
I think so. I think so. And for instance, from this point of view, we observed that the export rate that is crucial for buffering is one of those that are less heterogeneous across genes. It's very narrow distribution and low in efficiency. Which means that in this is a gate, somehow a gate that keeps the concentration of RNA in the cytoplasm, tries to keep the concentration of RNA in the cytoplasm constant. So I think that our results are indeed in line with this theory and this set of observations that several groups have obtained in the last years. Yes.
And did you use any drug to inhibit mRNA degradation?
No, not yet.
Okay, thank you very much.
Thank you very much.
And Mattia, you have another question in the chat.
Sure.
So can you?
Sure, I can read it. In the model, degradation is involved. Do you consider to implement mRNA silencing due to mRNA and long non-coding RNAs in the model to the model? This is a nice point, and generally speaking, the inclusion of direct inclusion of the effect of this kind of perturbations into the equations is interesting, of course. This can be rather complex and can reduce the generality of the model. That's the reason why I've not done it yet, but it could be done and could be interesting for specific research purposes in order to see if, in order to make a refined model which specifically accommodates a given experimental hypothesis. So this is something that could be done and could be interesting, but we're not planning to do it soon, honestly.
Okay, I guess there's no more question.
I have a quick question, if that's okay.
But who has one?
You mentioned that you want to couple these to proteomics, right? And so you already have 9 constants, if every single step. So how many more constants you are assuming will take? Because, once you get the polypeptide, you get the speed of the ribosomes, you get the degradation of the protein itself, and, et cetera, et cetera. So it's...
Sure. So theoretically speaking, you can accommodate as many variables as you can until the model is globally identifiable and you can deal with numerical issues. And this second point is not trivial. So this is the reason why creating reasonable simulated data is crucial for this kind of analysis. You have to be sure indeed that you can infer your kinetic rates. For instance, we tried to include nuclear decay in our model. And the model was globally identifiable. So theoretically speaking, you should be able to infer it. But with simulated data, we got very, very low correlations. And this is the reason why we decided to exclude this step, because we could not infer it, practically speaking. But going back to the proteomics, of course, you can think about reducing a little bit the complexity on the RNA side to increase the complexity of the protein side. That would be super interesting in my opinion. The nice thing with this kind of approach is that you can really tune what you do according to what you are interested in. That's the reason why we implement a simplified model, and you can do that again also with proteomics.
Anyway, very nice work. I really enjoy it, and nice for keeping it clean, as you call it, a simplified model, which was not that simple, but yeah, it was very nicely done. So thank you.
Thank you very much.
Okay, I guess we don't have any more question in the room. Well, kind of full. It looks like no, we don't have. So maybe we can end this webinar today. Thank you.
Thank
both
you again.
you,
Thank you,
Mattia,
everyone,
both
for the
Mattia.
opportunity. Thank you. Bye.
Thank you. Hope we see you next month once again. So thank you, both Matthias and Mattia. Matthias. Okay.
Bye.